Hello everyone, Assalamu alaikum. My name is Ahmed Shams. I am a lecturer of microbiology. This lecture is about penicillin binding proteins. I chose to start with this topic in a series of basic microbiology lectures because usually we don't study penicillin binding proteins in basic microbiology before we dive deep in other related topics. These topics may include the mechanism of action of beta-lactam antibiotics or the mechanism of resistance to beta-lactam antibiotics. But first of all, we should note that penicillin binding protein is a misleading name. This is for two reasons. The first one is that they don't bind penicillins only. They bind to all other beta-lactam antibiotics. The second reason is that binding to penicillins is not their main functions, not even the function they were designed for. So let's start and see what are penicillin binding proteins designed for. Our objectives behind this lecture will be three: the structure and types of penicillin binding proteins, mechanism of action of beta-lactams on penicillin binding proteins, and the relationship between beta-lactamases and penicillin binding proteins if any relationship is expected. And as you see, the core of our lecture is penicillin binding proteins, from which we will speak about the beta-lactamases and the beta-lactams and the relationship between all of them. Let's focus on this picture. I was happy to find this 3D diagram of a penicillin binding protein because when you imagine its position, especially in the cell wall of a gram-positive bacteria, you can easily recall its function. This green mountain is a penicillin binding protein extending from the cell membrane down and going upward through the peptidoglycan layer. This PG and the white arrow are pointing to a peptidoglycan layer that's being formed or woven by a penicillin binding protein. Please note that penicillin binding protein here is formed of two parts, the TP and GT part. We will go back to this picture, but I wanted you to imagine its 3D structure to make it easy to imagine its function. Before we dive deep in the structure of penicillin binding proteins as proteins, let's revise a principle in biochemistry. If you were an enzyme acting on a polypeptide chain, you certainly know that a polypeptide chain has two ends, carboxyl end and an amino end. Forget the amino end now, it's not significant in our lecture. But if you are an enzyme cutting through a polypeptide chain, if you are removing single amino acid or two amino acids at the carboxyl end, you are doing a carboxypeptidase action and function of penicillin. But if you are cutting in the middle of a polypeptide chain, removing more than two amino acids, three or more, you are doing every protein is formed of action. One polypeptide chain single amino acid and every polypeptide chain two amino acids formed of amino acids connected together. Carboxyl group that is a carboxy has two ends. Action. Removing more than two amino acids, three or more. When you cut by cutting in the middle of a polypeptide chain, or two you are doing an endopeptidase action. Close to the carboxyl group, you are doing a carboxypeptidase action. So when you cut one amino acid or two amino acids next to the carboxyl group, you are doing a carboxypeptidase action. But when you cut in the middle of the polypeptide chain, you are doing an endopeptidase action. So an endopeptidase action is cutting and releasing more than two amino acids, three or more. This is important to understand the mechanism of action of penicillin binding protein. And let's move to the next slide. This is a diagram of two layers of peptidoglycans. By definition, peptidoglycans are composed of peptides and glycans. Glycans means sugars. These sugars are alternating NAG and NAM arranged in layers. NAG stands for N-acetyl glucosamine and NAM stands for N-acetyl muramic acid. When you connect NAG and NAM, you are doing a glucosyl transferase activity. The peptide part of peptidoglycans is a polypeptide chain attached to each NAM. It's a pentapeptide of 5 amino acids. The terminal two amino acids are D-alanine. When you connect the D-alanine in the fourth position of a polypeptide chain to the amino acid in the third position of the opposite peptide chain, you are doing a transpeptidase activity, which is in broken line in brown. And this results in a cross-link. This cross-link is crucial in the stability of the peptidoglycan layer. If you break this cross-link, you are doing an endopeptidase activity. And as we mentioned in the last slide, if you cut in any position of these black arrows, you are doing also an endopeptidase action. You will set free, because we call it an endopeptidase action because you are setting 3 or more amino acids free. We mentioned also that if you remove 1 or 2 terminal amino acids at the carboxyl end, you are doing a carboxypeptidase activity, which is illustrated in red. After that introduction, now we can discuss the classification of penicillin binding proteins. They are classified into high molecular weight proteins and low molecular weight proteins according to their molecular weight. A bacterial cell wall usually have both types, but high molecular weight proteins are more important because they are responsible for the synthesis or building peptidoglycan layers. They do this by two mechanisms, the transpeptidase action and glucosyl transferase action. As we mentioned in the last slide, that glucosyl transferase action is when you connect two sugars together, is when you connect the NAM and NAG together. And the transpeptidase action is when you connect the polypeptide chains hanging from the NAM sugar together by a cross-link. And they are sub-classified into class A and class B. Class A are bifunctional. They can do the transpeptidase action and the glucosyl transferase action, while class B is monofunctional. They can do transpeptidase action only. Down to the left is a 2D diagram. It's showing that class A is composed of two parts or two domains, the transpeptidase upper domain and glucosyl transferase lower domain. And there is you can imagine that transpeptidase domain as a stapler, because a stapler can connect two papers together, and the transpeptidase domain is like a stapler that can connect two polypeptides together by a cross-link. And this red dot is what's called active site serine. It's an amino acid serine that is the active site where it can make the cross-link between the two polypeptide chains. While class B is composed only of transpeptidase domain. It has no glucosyl transferase action and no glucosyl transferase domain. And on the right side, you can see the low molecular weight proteins. You can think of them as a means of destruction. If the high molecular weight proteins are a means of construction, low molecular weight penicillin binding proteins are a means of destruction, because they can do the carboxypeptidase action by removing the terminal D-alanine or the terminal dipeptide D-alanine. And they can also do an endopeptidase action by breaking the cross-link, which is exactly the opposite action of the transpeptidase high molecular weight proteins. But there is no contradiction. They are working together because high molecular weight proteins are responsible for synthesis of peptidoglycan layer, and the low molecular weight proteins are responsible for remodeling and recycling. And to do remodeling and recycling, they have to break something. So there is no contradiction between two types of penicillin binding proteins. This slide is meant to explain the bifunctional activity of class A high molecular weight penicillin binding proteins. Here we have two layers of peptidoglycans and two polypeptide chains attached to the NAM saccharide, each composed of 5 amino acids. Terminal 2 are D-alanine. The amino acid in the third position is usually lysine in gram-positive bacteria and diamino picolinic acid in gram-negative bacteria. Anyway, this is a class A penicillin binding protein. It has two domains, transpeptidase domain and glucosyltransferase domain. The transpeptidase domain is responsible for making a cross-link between L-lysine and D-alanine in gram-positive. And the glucosyltransferase domain is responsible for connecting the sugars NAM and NAG together. We have just finished discussing the structure of penicillin binding proteins. And we did so for a purpose. This purpose is to answer this question. How do beta-lactams inhibit penicillin binding proteins? But to answer this question, we have to study or discuss how penicillin binding proteins actually work on a molecular level. So how a penicillin binding protein works on a molecular level. Please forget about the glucosyltransferase activity of penicillin binding proteins. Let's focus on transpeptidase activity, because this activity is what is inhibited by the beta-lactam antibiotics. So we are going to watch how transpeptidase activity is working in a penicillin binding protein. As we know, the pentapeptide chain attached to the NAM sugar has terminal D-alanine. When a penicillin binding protein with a transpeptidase activity meets this D-alanine, it attacks it with its active site serine. So an active site serine is an angry amino acid in the transpeptidase domain of penicillin binding proteins. It attacks D-alanine, removing one of the D-alanine, the terminal one, and forming an intermediate compound called the acyl D-alanine penicillin binding protein. This intermediate compound is further attacked by the lysine amino acid in the adjacent polypeptide chain attached to the opposite NAM sugar, removing or releasing the penicillin binding protein and forming the cross-link between the D-alanine of a polypeptide chain and the lysine of the other polypeptide chain. We have mentioned that beta-lactam antibiotics inactivate or inhibit penicillin binding proteins, specifically their transpeptidase domains. This is achieved by being a structural analog to D-alanine. We have said that once a penicillin binding protein meets a D-alanine, its angry active site serine attacks this D-alanine, forming a cross-link between one of the D-alanines and lysine or diamino picolinic acid in the opposite polypeptide chain. So what if this enzyme meets a beta-lactam ring disguised as D-alanine? And moreover, this beta-lactam ring chemically has greater affinity to the enzyme more than D-alanine. So definitely the enzyme will be deceived. It will attack the beta-lactam ring instead of D-alanine. But once it attacks the beta-lactam ring, it will be inactivated. So it can no longer do its function. And so there will be no cross-link formed, and the cell wall will be unstable and the cell may rupture. Let's watch this trap made by the beta-lactam ring to a penicillin binding protein. This is a beta-lactam ring, and on the right side is a D-alanine. You can see how similar they are. On the right side, you can see and watch the natural sequence of events. And a penicillin binding protein with its transpeptidase domain attacks this D-alanine. Then the cross-link is formed and the enzyme is released sound and safe. This is to the right. To the left, the enzyme is deceived. It attacks the beta-lactam ring, but instead of being released sound and safe, it's inactivated and no cross-link is formed. And you can think of the natural sequence as a stapler. A stapler can connect two papers together and still active to do its work again. But you can think of the beta-lactam ring with the penicillin binding protein as a stapler and a chewing gum. When a stapler attached to a chewing gum, it's inactivated and cannot do its action again. If you remember, our last objective of this lecture was to discuss and discover if there is a relationship between penicillin binding proteins and beta-lactamases. This might seem crazy, but serine beta-lactamases are thought to be evolved from penicillin binding proteins. In other words, beta-lactamases or serine beta-lactamases specifically were penicillin binding proteins that went crazy. It's like that bacteria have decided to have their revenge by transforming their penicillin binding proteins, which are inactivated by beta-lactam antibiotics, into zombie heads eating and damaging beta-lactams. So this is, I call it the revenge of bacteria. You can see on the left side a penicillin binding protein that's inactivated by a beta-lactam ring. But on the right side you can see a beta-lactamase enzyme that's released by a bacteria and has evolved from a penicillin binding protein that can destruct and damage the beta-lactam ring and render it inactive. Thank you. Shukran. I'm sorry, I forgot that I promised you to go back to this slide. We are here in this slide again to see how beta-lactam ring inhibit the transpeptidase domain of penicillin binding protein, and to know that there is an antibiotic that's not famous and not being used in the clinical field. It's called minomycin. Minomycin inhibit the glucosyl transferase activity or domain of penicillin binding protein, but this is not clinically important.